still has to go through the process of describing it. A formal description, yeah, will include comparisons against any similar species, but yeah, possibly 30 new candidate species up for description.
Yeah, and Tom mentions that many of the sponge collections could be potential new species as well from this expedition.
Yeah, sponges aren't a very well described group. And then Daniel from the Santa Barbara Natural History Museum is also saying there's a high likelihood of new species being discovered and eventually described from the sediment samples we're taking, because he's going to be looking at the micromollusks that live between the sediment grains, millimeters in size.
Micromollusks.
Yeah.
That's pretty amazing that some of our viewers can see maybe a new species to science right along with the scientists while you're watching. So there's a question from a high school student who says, you guys keep passing a large centipede looking thing and red fish. What are the names of those?
The centipede thing is a sea cucumber, holothurian. They look like they have lots of legs, but they're just small little feet that it uses to walk along the seafloor. They're not particularly agile. They just kind of sit and eat and ingest particulate matter from the seabed surface, where they extract the organic nutrients from it, and that's how they get on through life. But yeah, that's their mode of feeding. It's just deposit feeding. They're not particularly agile. They just kind of do their thing.
That's how they do.
And the red fish we're seeing, they look like they are long spine thorny heads. And if we get right now we're in transect, so we can't zoom in on it. But when we finish the transect, we can zoom in, and you can see if you follow the dorsal spines on the dorsal ridge, the third one back will be long, and that's where it's a distinguishing characteristic for that particular thorny head.
Fantastic. Here's a great question from Hong Kong. What is something you would love to come across while exploring? And this could be anything, not just living things.
A whale. That'd be great.
We did see dolphins this morning off the stern when we were launching, so we were hoping we'd be able to see them underwater. But
yeah, we get a lot of dolphins out here in the Channel Islands. Realistically,
I'd like to see sharks.
Yeah. Tom Leidig is saying a giant squid or a white shark.
Yes, I agree with that. Giant squid would be amazing. You guys have had some encounters with giant squid on some of your other expeditions, Nautilus, haven't you?
Yeah, Humboldt squid and large clouds of market squid, but no giants.
I think I would like to see a beaked whale for once on the seafloor.
For once.
You make in those trenches, because we've seen them like every single time I've been out since like the first time I was on Nautilus. We saw dozens of these beaked whale scours. We've never had any evidence of them actually doing it.
A little ground truthing, see it in action.
Yeah. How do they know that those whales are causing those marks if we never have any evidence?
Oh, good question.
There was a paper by another Corps of Exploration member, Lee Marsh, a few years ago, where they described patterns of beaked whale scours off the west coast, was it?
It was the Clarion-Clipperton fracture zone.
Yeah, CCZ. Yeah.
And Steve, I don't remember if they were able to determine how. I think they were actually comparing the size and shape of beaked whale heads to the scours found in the seafloor. That was one of the.
Yeah, it's
indicators.
2018 was the paper. I haven't read it in a while, so I need to go back and double check.
So did they tag them to know that they were going down as deep as the seafloor? Is that part of it?
Well, they can dive to several thousand meters, 2,000 or 3,000 meters.
But I mean, do they have like tags that tagged them going all the way down to the seafloor?
They must have data on that, yeah.
Just a comment for the viewers at home. There's a few people who are saying, I've refreshed and closed the browser out several times and the dot stays red. Just to all of you, I'm getting your comments. So go ahead and ask your question and we'll try to get it to the science team here.
The deepest whale scour in the CCZ was over 4,000 meters, 4,258 meters.
Wow.
Which is crazy.
Yeah.
A question for Samantha about how you became a navigator. Were you an intern first?
Ooh, that's a long winding road. How many meters left do we have for this transect ends? No, the short version is I actually started with the Ocean Exploration Trust as a Science Communication Fellow back in 2016. Science Communication Fellowship Program is a really awesome program bringing out informal and formal educators. So both classroom teachers, university teachers, museum or aquarium educators, and then even science writers, artists. It's a pretty unique program that brings folks out as expedition communicators. So Science Communication Fellows sit a watch in the control van. They bring in your questions just the way Laura is doing right now. They do live ship-to-shore interactions with schools and science centers all around the world. And so I came out first in that program and then actually ended up getting a position as doing social media for Ocean Exploration Trust, and I'm now the communications manager, managing our website, social media, press outreach, our video production team, and then I'm also often out on the ship as a communications lead, which is not a navigator at all. But with our reduced staffing for this year, due to COVID-19, an opportunity came out for a little bit of cross-training. So I am now learning what it takes to be a navigator, and it's been a really great learning opportunity, kind of combining all of what I've learned from both being out as communications lead, but also having now sat almost every seat in the control van for at least a few hours, if not for a few weeks. So kind of combining everything I've learned in those roles and using that situational awareness to help operations run smoothly as the communication point between the control van, the back deck operations, and the bridge where the captain and officers are moving the ship. That being said, we do have a navigation internship, but it's a partnership with the Coast Guard Academy and the Naval Academy. So that's a slightly different program. It's not a publicly open internship program the way our ROV engineering, video engineering, ocean science, and seafloor mapping programs are.
We've had some pretty diverse
Excellent.
people out as science communication fellows. We've had astronauts out here.
Mm-hmm.
What else have we had?
Yeah, I mean, we have...
She was a navigator.
Yeah, I
Was
think...
she?
Yeah,
Katie was
Katie
nav-
Coleman was a navigator.
Oh.
Yeah, but the fellow program is definitely grown. It originally started as a kind of formal classroom teacher program, bringing teachers to sea, and it's really expanded. We've had folks who have...
Black coral.
Oh.
Or
But we
sea
can't
pen,
stop.
no.
We're in a transect.
Yeah, no, sea pen.
Sea pen,
Just
okay.
correcting.
Are we still looking for sea pens to collect?
That would be for Taylor Ann, and she's coming on in 5 minutes.
Oh, okay,
so
great.
she can answer that.
And even
Yeah.
better, our transect is ending around the same time, so that's perfect.
So there, speaking of transects, there are a lot of questions in the chat box about what is a transect and what type of data is collected and how it's used.
Well, this transect is complete, so I will finish that and then let the science team take over what that means. And science, do we just want to look around here while we wait for the next watch to come on in 5 minutes?
Yeah, I think that sounds like a good idea. Lizzie stepped away, but that seems like since we're kind of getting near the transition point, sounds good to me.
Yeah, I want to zoom in for the student who asked about rockfish. Al, let's zoom in.
Oh, good. And a transect is really, it's following a line, some sort of a heading, as in the case with the ROV, or it could be a physical.
Yeah.
Oh, there we go. Hold on, I'll answer the transect question in just a sec. Let's zoom in.
So is this a short spine or a long spine, Thorny Head? Looks
Long spine.
long.
You can zoom in further.
Yep. All right, long spine.
So yeah, the third. Yep, it looks like it's got a long spine. So it's kind of a hard angle because it's sort of looking kind of straight down on it. But you can see that one of the spines is definitely longer than the rest. Oop. And whereas if it was a short spine thorny head, then you wouldn't see that pronounced elongated spine. There it is sticking, jutting up there. And so we just finished this transect. And so what that means is we were following a certain bearing and going a certain distance. In this case, how far did we travel on this transect? Did we go 200 meters?
200 meters, yep.
Yeah. And we keep it at a consistent speed. So like
I think we should
0.5 knots.
should get sorted out.
And
here.
that's how we do transects in
Turning
deep
over
water
a mess.
environments. In shallow water environments, you can do transects by pulling a meter tape out and along a certain heading or bearing, and then you can go along that transect and record species. So as we're going along the transect, we're taking note of what we're seeing. And so in this, we're just trying to characterize the deep water environment here. So it's soft sediment. It's got the brittle stars or orpheroids. We've got the sea cucumbers, holothurians. We've got the long spine thorny heads, and we've seen some gastropods. So we're taking notes on all this as we're working our way along the transect. Sometimes when you work on different transects, you're looking for specific, maybe just fish, or you're looking for a target species. But a lot of times when you do transects, you're trying to characterize everything along it. And it's a way to sort of have an unbiased observation of an area. Instead of picking out some hot spots that look good, you just sort of run a transect and record what you see. And it creates a baseline over time to sort of help us understand different areas and how diverse they are.
That's great. And along those lines, what are the two green dots that people are seeing when we're doing a transect? And like, how would you measure some of the animals you're seeing?
That's a great, well, we just turned them off, but when we're doing the transect, it helps for scale, because especially when you're not down there and sometimes trying to understand how big something is, it's hard to know looking at a picture. So for the remotely operated vehicles, the ROVs, they have these laser pointers, and they're a certain distance apart, I think 10 centimeters apart. So you can use it as a reference point to be able to estimate sizes of fish. In this case, we're also looking at sponges and sea fans and corals, and so it helps us to provide scale.
Alrighty, so our watch changes.
Snakehead eel pout. Oh yeah, watch change.
Right there in that. Oh, let's zoom in on that.
Yeah, and you can see his scales, the iridescent scales under his eye and on his pectoral fin from here.
It looks white to me.
Mm-hmm.
That's
Yeah,
really
it's a little
neat.
blown out, but once you...
Nice.
Beautiful. It's a cool fish.
It really is. And I appreciate it not darting off. So you get a good look at it.
Yeah, you can see the scales on the face, and its fin are much more reflective than the rest of its body. So that's a good indicator that this is a snakehead eel pout.
And so snakehead eel pouts are deposit feeders as well. They're feeding on just being opportunistic, I would imagine. And they use that long, slender, their tail, they can actually create, just cover themselves with the soft sediment.
They are predatory, consuming microscopic zooplankton and crustaceans, juvenile fish, insects, well, not insects like arthropods probably, and crustaceans as adults.
Thanks so much for answering the questions today. I know you're on a watch shift, so I'm going to jump off so others can get on, but really, really appreciate answering these questions, and we'll be back at 3 o'clock today with more.
Thank you,
Thanks so
Laura.
much for joining,
that was
Laura.
Fun.
Yeah. All right, so as Laura mentioned, we are changing shifts. So Julie and I will exit off of the V-Link software so we can have the next watch shift come in and get up to speed. The pilots, the navigator, the video, and the data logger are also changing shifts as well. So you'll hear new voices come on and you'll hear ours leave, but we had a great dive and we hope you'll continue to join us throughout our cruise.
Yes, thank you very much.
And I'll see you tonight, Julie. Or I'll hear
see you
you
tonight at 8 o'clock. Yeah, definitely. Yeah. All right.
All right, bye.
Have a good rest of the day. Bye-bye.
Hello. I'm just going to do a quick DVL reset before you get going. I see you're out of auto XY now. Okay. Who do we have on shore? Or maybe they're still handing over.
Yeah. Hey,
Hello.
Shore is joining now. We've been just on Nautilus channel. This is Meredith.
Hey, Meredith.
So,
this is Abby.
Hello, Abby.
Hi, Nautilus.
Oh.
yep.
It is I, the voice of Nautilus.
Well, we're starting to recognize your individual voices, but I think it's harder being from shore.
Well, you keep switching watches too. So.
Yep, that's true. Yep.
All right, so we're on our way to BG, as I understand it.
Yep.
Okay. We'll keep going at 045.
045.
Bridge nav. Step 200 meters, bearing 045, speed 0.3. Thank you. And we'll speed up, Jake, if we think it's
Yeah, that's
warranted.
sounds
But
good.
we'll start there.
So we've traditionally on this watch started with a round of introductions right after the watch change. So I'll go ahead and get started. This is Meredith Everett. I'm a geneticist at the Northwest Fisheries Science Center, where I work on the genetics and taxonomy of deep sea corals and increasingly sponges as for the Deep Sea Coral Research and Technology Program, who is currently in the wrap-up year of their West Coast Research Initiative.
I can go next. I'm Abby. I'm on Site today. I'm a contractor also at the Northwest Fisheries Science Center based up in Seattle. And I work for a team run by Liz Clark that has an AUV called Popoki. And my background is sponge ecology, so I've been keeping an eye on the sponge collections during this cruise in the Channel Islands.
This is Renato Kane. I'm in the navigator seat.
This is Stephen Matter in the video chair.
Dan in the Argus chair.
And Jake in the Herc chair.
And Taylor Ann in the data log chair.
Excellent. Thank you all. So I'm looking at the screen here. Is it actually this steep, or is it just kind of an illusion? We tilted down a little.
What's your camera angle at?
Somewhat steep.
Okay.
Down a little bit.
Down a little bit, yeah.
It's a bit steep. Do we want the lasers on as we're going?
Yeah, if we could get the lasers on, that would be great.
Why is the aft LED off on the ground fault?
Looks like Argus is just starting to move.
Okay.
Roger.
Lizzie's commenting from shore that it should be pretty steep right here. It may flatten out towards BG. So, okay.
Rumor is there was a cliff on this dive.
So far, I think that's been all rumor, at least that I've seen.
The most cliff-like
Unless
thing
we're...
I see is between BH and BI. There's this kind of steeper ridge, what appears to be a steeper ridge in the data, but may be the same as the slope surrounding.
Yeah. Well, it doesn't need to be a cliff as long as we eventually find some hard substrate.
I'll pull up, that's the slope map for it. So I think I set the red to be 50-degree slope.
Yeah. That looks similar to what we have in the dive plan. So far this dive, it's been largely this soft bottom habitat with, I think, one or two scattered rocks with individual corals or sponges on them. Seen a lot of holothurians.
We're quite deep. So we could potentially see some interesting deeper species that we find on the soft sediment substrates. I know we came across some of those sponges that get called the fiber optic sponges because they have a huge long stalk of glass spicules, and then they look like a big kind of orange tulip last time we were south of Santa Cruz Island.
Yeah, those were fun. You can end up in a whole field of them, and it's like being in this like crazy underwater oversized tulip garden.
That's surreal.
So Lizzie is asking if we can put the slope back up on HiPAC and talk about what we're seeing and where we think we'll encounter hard habitat. Because apparently your slope map is way better than hers.
Oh, the slope. Got it. I thought you said slurp.
Yeah.
I was like, why do you want to look at the slurp chart?
Also,
What
I
is
thought
the
you
slope
said
map?
Sure, yeah. We can have the slope map up. I also have backscatter as well if we want to take a look at that, but I think that's pretty well planned out. Once we go
Yeah.
from BG to BG1, should be some.
Yeah. So Lizzie, I
back
know
to
you're
slope.
listening.
So I can zoom out, and as we're going up, we can
Yeah.
look at it.
Lizzie says, I guess my question is, do we need BG?
It's there's like, this is the other side of the slope of this feature, but if we're not seeing anything that we need to see any, we could just go straight up north and then go to BG1, depending on if you think there's going to be something over there that's different from over here.
Lizzie said things like she's wondering if it may just turn out to be a wild goose chase waypoint, because yeah, I don't know if we're going to see anything different over there than we will see straight up. On the other hand, it is a long dive. Yeah, she agrees we won't see anything different. So why don't we go ahead and adjust the move and just go straight north and kind of catch the contours that we'll see BG1 on.
Bridge nav.
Gonna strafe your gauges there.
Utter.
Can we change course to north? Thank you.
5, 7, 6, 8. 4, 5, 6, 7, 8. Is that right?
Sounds right.
On previous dives around South Santa Cruz Island, it's rare that we've gotten this deep, so it's really nice to explore this deeper habitat. And hopefully we'll get some hard bottom here soon.
Are there any corals we're particularly looking out for today, Meredith?
Let's see, down here, down this deep, some of the blacks probably. Looking at our collection target list, we've been able to collect, yesterday we were able to collect another Amphiphila, which is nice. That's a particularly tricky group to collect because they tend to be really low to the rock. Looking for possibly more Plexaurids, either Thesia or Swift or Placagorgia, and then any of the Pernoids we see. I have seen one sea pen so far. Yeah, seen one sea pen so far since watch change here. Oh, there's a nice hagfish there, and it looks like it's the Stachytilum species.
And are these lighter coloured transparent ones Panicia? Or do you think they're all the same?
I think they are probably Panicia, although I'm not as good on my holothurians as
hollows. Little jellies, maybe Paraphyla?
There's definitely been some Paraphyla. And a lot of these nice little young thorny heads scattered everywhere. So far they've all been long spine thorny heads. So do we have any specific sponge targets?
Well, I was having a look at what we collected last time south of here, and we did come across some of what the Southwest Centre they nicknamed the light bulb sponge, which is a glass sponge that looks like lots of little tubes, and the family treated it today. and that is one of the sponges on our collection list. So we'll be keeping an eye out for that. But that is definitely needs some hard substrate. So yeah, that's something to look out for if we get to any ridges or cliffs.
Check it out.
Excellent.
Yeah.
My video is very jumpy today, so I'm just doing a quick check to see if I've got any tabs open, taking up bandwidth.
Mine was a little jumpy this morning, but seems to be pretty stable now, so. Earlier in this dive, we had some of these Panicia doing the thing where they sit up with their heads up in the water column. I'm hoping we get a little more of that.
All right.
I've definitely, so I remember seeing a lot of that, like you'd find big groups of sea cucumbers doing that when scuba diving in coral reef habitats, and I always thought it was something related to spawning. So it's quite an interesting behavior. We should have a little zoom on and.
First, we have to find one doing it.
Yep.
Starting to see a few sea pens around, which we haven't seen many of this dive so far. Seeing the species as yet to be determined that last year successfully ran away from the ROV 2 out of 3 times. And then the Stachytilum.
Oh, these are the ones, they look like little white transparent feathers.
Yeah.
The little, yes, yes. So I think they only just finished the transect, so we probably need to make some distance before we start another one.
Yeah. We're 120 meters from the end of the last one.
Oh.
Oh. Well, in that case, we can go ahead and start another transect.
Sure. You guys all good with that?
Sure.
All right, Taylor Ann.
All right. I have that as transect number 3.
3. Aye.
Want to go autos there.
You said I want to go autos?
What's that?
I want to go autos?
You all want to go autos for the transect? Auto out at least.
Yep, in auto out.
It is auto out?
Yeah, it's on auto out.
I always think that starting the transect is a really great way to get some, hard bottom habitat of things, because then you're on transect and can't stop to collect anything. So.
Yep.
So yeah.
Always the way.
I also like to provide Tom with many things that are very easy for him to count.
I did see some little eel pouts earlier. Might be something we could get a zoom on when we're off transect.
Yeah, we've been seeing snakehead eel pouts, the ones with the really shiny faces.
Oh, with the iridescence. Yes.
Yeah. And there have been lots of these little fish that have been kind of diving through the camera view, some of which I think are that eel pout that hangs out up in the water column and will get confused by the lights, and some of which I think have been bristlemouths.
So Meredith, what were those? Are they called penatula, the little sea pens?
The ones we have, I've been seeing like the little white ones. I'm not sure if those are penatula or if those are possibly a funiculina or something else. They were something that when we collected one last year, I didn't recognize what it was once we got it up to the surface. And it's one that will be being shipped off to Gary Williams probably next week.
I'm going to call it small sea pens.
Small sea pens, yeah.
So it doesn't look like. Yeah,
go ahead.
I was going to say, it doesn't look like there's been any niskins so far.
Nope, no niskins so far. So we should fire one at the end of this transect. This is a new method I've been trying in the last year. Previously, we've just tried to fire niskins in a mix of habitats, both with corals and without. And since on some of these cruises we've been doing the quantitative transects, like the one we're on now, I've been trying to pair niskins to either the start or end of a transect so that it can be matched to what we see on the transect. And see how those two methods line up.
I mean, you'd think you'd have a good chance because the transects are 200 meters, so should pick up this. That's about the range that you'll get in the eDNA sample, isn't it?
Yeah, probably a little less than 200 meters in the eDNA sample. Closer to, we usually say about 100.
Right.
You know, the nice thing would be infinite Niskin bottles where we could, you know, fire one beginning, middle, and end, but that's somewhat impractical. So this way, at least for a portion of the transect, presumably will be matched with the eDNA. Given other variables like current, etc., we found sometimes in high current environments that you don't really necessarily match what you're seeing on the seafloor to what you're getting in the sample, largely because in high current areas, the water you're getting is not the local water.
Yeah, it's always a big unknown, and it is something we don't have a great way to measure at the moment. When we're doing cruises like this, I know with some methods you can use to survey fishes is by using a camera with bait attached, and then you kind of drop a camera, and you can measure. You get video images or still images of the fish that come and feed on the bait, but determining which area you're sampling is quite difficult because, yeah, depending on the current, that bait plume could be staying fairly local or traveling quite some distances. So there's all these variables to take into account that can be quite difficult to quantify.
Yeah.
Still, eDNA will definitely give us that wider view than the few meters that we get in front of us from Herc's camera.
Yeah, we like to call it a different size net.
Yep.
This little star there.
Oh, I've got channel 3 at the moment. That's quite nice. Have a look at what what they're saying on the bridge. View of the island.
Sounds like you might have quite a delay, Abigail.
Oh,
Um,
I'll refresh
yeah,
everything.
give it a shot. It was
Sorry,
a couple
an
minutes
hour
ago. I
ago.
switched it a couple minutes ago.
The people at home ignore that. It's still there. What's going on?
Is it paused on the bridge?
Oh no, now I've got high pack. There we go. I better check the other one too in case I'm back in time on channel 1.
Just on the top of the screen, there's one little blip of possible rock sticking out of the mud. Covered in mud, so there's nothing really.
I was going to say, I don't know how I'll know because I'm still seeing mud.
The next interesting thing that comes in the frame, we'll tell you, and then you guys have to tell us when you see it.
Yeah. That sounds good. We've got 4 hours.
All right, there's a
6 p.m. tonight.
sea cucumbers in the top left, maybe a hagfish.
Yes, I see that.
Okay, great.
Great.
Perfect.
We're lined up. We're synced.
I am interested in the satellite feed 3. So if you want to look at channel 3, I'm going to switch it. I'll switch it back to the bow cam really quickly.
Okay.
And I just switched it. Let me know when you see the bow cam.
I see it now.
I see it now.
Okay, not too bad. A few seconds. That's what we expect. So seems like we're back in sync.
Excellent. Thank you.
Oh, now there on the bottom of the screen is a sea pen that I do think is a Penatula. Those ones are super fun because when you see them on the seafloor, they're not a big sea pen, but they'll be the full like 10 centimeters high, pretty fluffy. And when you get them on the surface, they are these tiny little 1-inch sticks with little tiny like fronds because they just expel all the water out of their tissues and shrink by 5 or 6 times. I'm always kind of amazed when they get them in the trawl survey because it's like, how did this stay in the net?
Yeah, you'd think it must have been trapped in mud or something, or
Yeah,
I find it even more amazing when they get beautiful coral. Like sometimes those Gorgonian a little fan will come up almost intact, and you can't help thinking like, how did that not get crushed by rocks or something?
And the fans are pretty robust because they're pretty flexible.
Just an illusion.
Yes, they're actually pretty tough. It's when they get like big pieces of bamboo coral that still have tissue on them that I find slightly more amazing. All right, I'm going to pop out for my quick meeting and leave Abby here. I will be back within probably 25 minutes.
All right, have a good meeting. Uh-oh, Nautilus, buckle up. Got only one scientist ashore. Parelia Jelly, feel free to comment on what we're seeing. I know that it does help the analysts. Some more thorny heads. Taylor Ann, have we taken any samples for your project yet this today?
Not today, I don't think. Nope, we only have 2 samples and a push core. I think we got some snails.
I think I saw that, yeah. So what's your main target for looking for the microplastics? I've seen that we've been collecting some kelp, some holothurians. Are there any groups in particular?
Yeah, so I initially was thinking of sea pens, but hadn't seen very many like we thought we were gonna see when we were first starting these dives. So then I started to go for sea urchins because I wanted to
do it again. I got stuck on stupid.
at least have some like detritivore, something feeding in the sediment. Because I also was thinking of sea pigs in the beginning, because I wanted to get to see the difference in if there was the difference in ingestion of microplastics from like water column feeders or organisms that feed in the sediment. But my main, I guess, goal was to see how the concentrations and types of microplastics changed as a function of water mass properties associated with depths. So taking Niskins and filtering those out, and then tying that into the biological community and seeing as we change from sites closer to shore, farther from shore, if we see organisms ingesting different concentrations or amounts of microplastics, and if so, which types of microplastics are the ones that they're ingesting, and which ones are making it all the way down to the deep sea. So I have some shallow sites and some deeper sites, and then some sites near shore and farther offshore. So we'll see how that changes as a function of distance from shore, depth, water mass properties. So the only thing that kind of I wish I could do that I can't is have more of a physical component, tying in like physics, using like an ADCP and getting the flow rates and the current of what's going on in the water column, because that really affects how microplastics move and potentially settle as well. But that's something that I'll be doing in my kelp study, which is why I'm collecting kelp blades. So I'm doing the same project in kelp forests to see how the flow of water, how that changes in kelp forests. It slows down due to the presence of thick canopies of kelp, and seeing if that can then cause these kelp forests to be hotspots for microplastics to accumulate, since water sits in one area for a longer period of time. Potentially these microplastics could be too. Yeah, so seeing if kelp can actually be a vector for microplastics. So traveling from the surface waters all the way down to the deep sea, if it could be carrying along plastics or anything on its surface, specifically looking at Macrocystis porifera, which is,
yeah. It's super interesting.
Yeah, we'll see.
I remember, yeah. I'm really curious to see what you find. I've heard that I saw some a modeling project where they were modeling kind of plastic particles on the ocean and came up with, I mean, it seemed like a huge number, but they were saying it's just a fraction, because really most of it is ending up down in the deep sea. So it's kind of an invisible, it's an invisible problem. Things just eventually will just sink. So it's really cool to be looking at some of these deposit feeders. I'm curious, do you do chemical analysis to find the plastics, or is it kind of, are they large enough that you could do like microscopy?
Yeah, so I'll be using a micro FTIR, which uses like infrared technology. And there's like a library on that machine that can identify the different types of plastics. So all of the information of the different types of plastics that are out there is stored on that machine. And I'll run my filters underneath that machine and let it analyze it and tell me what we're seeing. So then
Fantastic.
I'll do counts, and then I'll be able to know what types we're seeing too.
Well, definitely give us a shout. I think after this transit, while we're still in kind of these soft bottom habitats, is a good opportunity to do any collections if we see some kelp.
Yeah, sure.
Yeah. So I'm still just seeing little tiny thorny heads, more of the sea cucumbers.
Are those a different species of rockfish, or are they that are smaller, or are they just juveniles?
They're just
I'm just going off, yeah, what Meredith was saying, that I think at this depth she was saying we'd been seeing the long spines.
Mm.
I do know that the little ones tend to be kind of darker grey rather than bright red. Yep, we have Tom Leidick from the Southwest Fisheries Science Centre on the science chat saying, yep, just juvies, baby long spines. Should probably put that out there. Any of the folks on the science chat at the moment, there is a free spot on VLink. Open invitation to join in this mud dive. Looks like we're not far from the end of our transit now.
Yep, about 40 meters left. Some kind of soul, maybe just out of frame. Thought I saw
His nose poking out.
Yeah.
Was it a petrale? I imagine everybody's pretty good at the petrale souls after the other night's dive.
Yeah, I couldn't quite tell. It was only just the head.
Just oh, this is a good tip from Tom. So the short spine thorny heads, you tend to find the largest individuals at the deepest depth. So if we see a short spine, it's going to be a big fish down here because we're still over 1,000 meters.
Mm.
Something we see not just with fish but invertebrates as well, as you often have zonation by depth. But it's really just because all the environmental factors like pressure and temperature change as you increase the depth. So the way we see that in our species counts, as you see a change in which assemblages, which species are present along different depth zones. Two really important factors, a depth and then also obviously habitat type. So you see a whole different complement of species on these sandy habitats that we do when we see some of the more vertical cliffs or around kind of boulders and hard substrates. Well, apparently that little flash of the flatfish was enough for Tom to ID it. He said it was a Dover sole.
A Dover sole?
Yep. Very tiny mouth. I think that's the one actually where the Latin name is Microstomus pacificus, which actually might even mean tiny mouth. So I have quite a lot of family that have been watching along the dives, and of all the deep sea images and things that I've been sending them or that Nautilus has been tweeting out, the one that they've sent recently is about the owl. I'm curious if any of you saw that owl on deck.
2 owls.
2 owls.
No, we
That's
were, I
crazy.
think, sleeping when that was going on.
Hmm.
This watch was. So it was probably
Getting
during
some rest.
reasonable hours of the day.
Yeah.
Yeah, 12 to 4 is tough. I struggled a bit the next day after that Petrali Soul dive. Is it a random process, the way that they assign watches on the shift, or do you get some say on which shifts you get assigned to?
Part of it is pairing teams well. So some people who are experienced, some people who are less experienced, and then it just so
Right.
Whichever watch it so happens to fall on. That's kind of, if someone has a strong preference and it's a lateral move and it's not going to affect anything, then sometimes they can accommodate that.
Mm-hmm. So kind of matching up skill sets and things.
Yeah.
Yeah, makes sense.
All right, that's the end of transect.
All right.
We can still keep heading north and then eventually go west towards BG1, unless you'd like to get there at a different angle.
I think we keep heading north for the minute.
Sure.
Because there's nothing particularly standing out to me on this contour, so I think, yep, we keep going.
Yeah, sounds good.
Let's transit. Oh, I know what we need to do. Take a Niskin.
Oh, yes.
We've just been
Oh.
talking about it. Yes, if now's a good time,
Niskin?
then I think we should do it. Yep.
Do you want me to stop up, or are you okay doing it on the fly?
Utter.
Okay.